We all start life as one single cell. Then that cell divides and we're two cells, then four, then eight. Cells form tissues, tissues form organs, organs form us. These cell divisions, by which we go from a single cell to 100 trillion cells, are called growth. And growth seems like a simple thing because when we think of it, we typically think of someone getting taller, or later in life, wider. But to cells, growth isn't simple. Cell division is an intricate chemical dance that's part individual, part community-driven. And in a neighborhood of 100 trillion cells, sometimes things go wrong. Maybe an individual cell's set of instructions, or DNA, gets a typo, what we call a mutation. Most of the time the cell senses mistakes and shuts itself down, or the system detects a troublemaker and eliminates it. But enough mutations can bypass these fail-safes, driving the cell to divide recklessly. That one rogue cell becomes 2, then 4, then 8. At every stage, the incorrect instructions are passed along to the cell's offspring. Weeks, months, or years after that one rogue cell transformed, you might see your doctor about a lump in your breast. Difficulty going to the bathroom could reveal a problem in your intestine, prostate, or bladder. Or a routine blood test might count too many white cells or elevated liver enzymes. Your doctor delivers the bad news. It's cancer. From here, your strategy will depend on where the cancer is and how far it's progressed. If the tumor is slow-growing and in one place, surgery might be all you need, if anything. If the tumor is fast-growing or invading nearby tissue, your doctor might recommend radiation or surgery followed by radiation. If the cancer has spread, or if it's inherently everywhere like a leukemia, your doctor will most likely recommend chemotherapy, or a combination of radiation and chemo. Radiation and most forms of chemo work by physically shredding the cell's DNA, or disrupting the copying machinery. But neither radiation nor chemotherapeutic drugs target only cancer cells. Radiation hits whatever you point it at, and your bloodstream carries chemotherapeutics all over your body. So what happens when different cells get hit? Let's look at a healthy liver cell, a healthy hair cell, and a cancerous cell. The healthy liver cell divides only when it's stressed. The healthy hair cell divides frequently, and the cancer cell divides even more frequently and recklessly. When you take a chemotherapeutic drug, it will hit all of these cells. And remember that the drugs work typically by disrupting cell division, so every time a cell divides, it opens itself up to attack. And that means that the more frequently a cell divides, the more likely the drug is to kill it. So remember that hair cell? It divides frequently and isn't a threat. And there are other frequently dividing cells in your body like skin cells, gut cells, and blood cells. So the list of unpleasant side effects of cancer treatment parallels these tissue types: hair loss, skin rashes, nausea, vomiting, fatigue, weight loss, and pain. That makes sense because these are the cells that get hit the hardest. So in the end, it's all about growth. Cancer hijacks cells' natural division machinery and forces them to put the pedal to the metal, growing rapidly and recklessly. But using chemotherapeutic drugs, we take advantage of that aggressiveness, and we turn cancer's main strength into a weakness.
On September 1st, 1953, William Scoville used a hand crank and a cheap drill saw to bore into a young man's skull, cutting away vital pieces of his brain and sucking them out through a metal tube. But this wasn't a scene from a horror film or a gruesome police report. Dr. Scoville was one of the most renowned neurosurgeons of his time, and the young man was Henry Molaison, the famous patient known as HM, whose case provided amazing insights into how our brains work. As a boy, Henry had cracked his skull in an accident and soon began having seizures, blacking out, and losing control of bodily functions. After enduring years of frequent episodes and even dropping out of high school, the desperate young man had turned to Dr. Scoville, a daredevil known for risky surgeries. Partial lobotomies had been used for decades to treat mental patients, based on the notion that mental functions were strictly localized to corresponding brain areas. Having successfully used hippocampus, a part of the limbic system that was associated with emotion, but whose function was unknown. At first glance, the operation had succeeded. HM appeared, with no change in personality, and his IQ even improved. But there was one problem. His memory was shot. Besides losing most of his memories from the previous decade, HM was unable to form new ones, forgetting what day it was, repeating comments, and even eating multiple meals in a row. When Scoville informed another expert, Wilder Penfield, of the results, he sent a PhD student named Brenda Milner to study HM at his parents' home, where he now spent his days doing odd chores and watching classic movies for the first time, over and over. What she discovered through a series of tests and interviews didn't just contribute greatly to the study of memory. It redefined what memory even meant. One of Milner's findings shed light on the obvious fact that although HM couldn't form new memories, he still retained information long enough from moment to moment to finish a bathroom. When Milner gave him a random number, he managed to remember it for 15 minutes by repeating it to himself constantly. But only 5 minutes later, he forgot the test had even taken place. Neuroscientists had thought of memory as monolithic, all of it essentially the same, and stored throughout the brain. Milner's results were not only the first clue for the now familiar distinction between short-term and long-term memory, but showed that each uses different brain regions. We now know that memory formation involves several steps. After immediate sensory data is temporarily transcribed by neurons in the cortex, it travels to the hippocampus, where special proteins work to strengthen the cortical synaptic connections. If the experience was strong enough, or we recall it periodically in the first few days, the hippocampus then transfers the memory back to the cortex for permanent storage. HM initial impressions, but without a hippocampus to perform this memory consolidation, they eroded like messages scrawled in sand. But this was not the only memory distinction Milner found. In this experiment, she asked HM to trace a third star in the narrow space between the outlines of two concentric ones, while he could only see his paper and pencil through a mirror. Like anyone else performing such an awkward task for the first time, he did horribly. But surprisingly, he improved over repeated trials, even though he had no memory of previous attempts. His unconscious motor centers remembered what the conscious mind had forgotten. What Milner had discovered was that the declarative memory of names, dates, and facts is different from the procedural memory of riding a bicycle or signing your name. And we now know that procedural memory relies more on the basal ganglia and cerebellum, structures that were intact in HM's brain. This distinction between knowing that and knowing how has underpinned all memory research since. HM died at the age of 82 after a mostly peaceful life in a nursing home. Over the years, he had been examined by more than 100 neuroscientists, making his the most studied mind in history. Upon his death, his brain was preserved and scanned before being cut into over 2,000 individual slices and photographed to form a digital map down to the level of individual neurons, all in a live broadcast watched by 400,000 people. Though HM spent most of his life forgetting things, he and his contributions to our understanding of memory will be remembered for generations to come.